Hey everybody, this is our lesson on the origins of life, early Earth, and the theory of evolution. Um, just as a disclaimer, I'm sorry if you hear my dishwasher in the background. But we are looking basically at evolution and immunity in this unit. So, one definition to make sure we are clear on is the definition of a theory. So, a theory is a well substantiated explanation for some aspect of the natural world that can incorporate facts, laws, inferences and tested hypotheses. So, these can be discarded as new information is gained. This is not a law. So, laws are proven ideas in science. But some examples of theories that have been in a certain regard accepted in the history of science are the theory of evolution by natural selection, cell theory, and the theory of disease. So, theory, according to Merriam-Webster, so just dictionary definition, is a plausible or scientifically acceptable general principle or body of principles offered to explain some phenomena. It's also defined as a hypothesis assumed for the sake of argument or investigation, and it's an unproved assumption, but there is evidence that it is possible. So it could be one of the potential explanations of some phenomena. So now let's get into the origin of life. So can living things arise from non-living things? We do have one definition to know, and that's called spontaneous generation. So this is the living organisms arriving from non-living matter. We can also refer to this as abiogenesis. And this was tested by three scientists. So Francesco Redi, Lazzaro Spallanzani, and Louis Pasteur, which we have heard of that one if you've heard of pasteurization. So it says you can review these from Unit 3. You can look back on a little bit of stuff there, but we don't have as much. So first, Francesco Redi, his experiment was performed in the 1600s, and he studied maggots that were thought to come from rotting meat. So he would have samples of meat in a jar, and some of them were open, some of them were closed, and some of them had fully closed, and then some of them had cheesecloth over them, which is a breathable fabric. And he tried to determine if maggots came from dead, rotting meat. Okay. So the controlled experiment was an uncovered jar containing meat and jars with meat covered with a net, like I said, some sort of cheesecloth. Air could enter, but flies could not. They only could in the uncovered jars. And the conclusion was maggots came from the eggs that were laid by the flies, not from the dead meat or the rotting meat itself. Oh, my goodness. So here's an example of Lazzaro I can't do it every time. Spallanzani, where he did a similar experiment, except he tried to see if microorganisms could arise from some sort of broth or liquid that would be heated, an abiogenetic generation. So as you could see, growth would occur as long as the flask was open. So air had to be an option. Organisms in the air had to be able to get into the system. If you've ever made a petri dish before, which we had this issue in the AP biology class a couple years ago, you have to make them and close them as soon as possible to try to prevent any bacteria or any germs in the air from getting on the plate and growing. Otherwise, you can't use the plate. So you have to have them as sterile plates. But if they aren't closed quick enough, very little microorganisms can get in there and continue to grow, and then you'll see that growth. You can't use the plate anymore. So Spallanzani's experiment was performed in the 1700s. And again, like I said, studied microorganisms using a new tool. He actually would use the microscope to be able to see these. He used broth in sealed and unsealed containers, and the broth that was heated and sealed did not spoil, but the one that was unsealed would spoil after heating. And then Louis Pasteur, we talked about pasteurization of milk, and he looked at, again, you have to have access to the liquid to see if growth would occur. And his was performed in the 1800s, so this is 200 years worth of experiments that were trying to study this. And he used the swan-necked flask, which is what you see in this picture. And that had no stopper, and he would boil the broth. The curved neck would trap dust and particles before it could get into the solution. So that's why this was an effective tool to use. And the conclusion was that the broth didn't spoil when the microbes could not reach it. So he disproved spontaneous generation by this test. So now that we know what abiogenesis is, we can talk about biogenesis, which is living organisms coming from other living organisms. If life only comes from other life, how did life begin? So, the formation of the Earth about 5 billion years ago, the solar system was a swirling mass of gas and dust. It collapsed inward and formed the sun, and then the Earth is dated via, carbon dating, things like that, about 4.6 billion years old. So if life comes only from other life, how did it begin? And we have to understand what the atmospheric conditions were at this point to know what could have occurred here. So first off, the most common gas that was present in the atmosphere at this time was hydrogen, but also water vapor, ammonia, and methane were present. And the conditions were very unstable due to a bunch of different issues, but volcanic eruptions was a huge one. And then meteorite bombardment. So there were lots of, debris from the atmosphere coming down and hitting the Earth. This was common because we didn't have magnetic poles and protection because there was no atmosphere that we have today. The Earth was very, very hot at this time, and the temperatures were above boiling. As it cooled, the water vapor condensed and formed pools, so, just like puddles form in the ground. H2 gas would escape. Ammonia would decompose into N2 and H2. And the first life that was present on Earth were prokaryotic, so bacteria. And this was determined by examining fossils. So there are 4 requirements for the origin of life. And the first one is the formation of simple organic compounds. This was studied by Alexander Oparin. And he, explained the 4 major components of early atmosphere. And that the high temperatures and energy from lightning caused molecules to split. They recombined to form new molecules and condense, which settled in those pools. You may have heard of this called the primordial soup or organic soup. This was, he hypothesized this, that chemical reactions form proteins and organic compounds, but he did not test this. Okay, so this is an untested hypothesis. Until 1953, where Stanley Miller and Harold Urey tested this hypothesis, they were able to form the amino acids alanine and glycine. And this is the process that they used to do that. So you can pause it and read over those. The second requirement for the origin of life is the formation of complex organic compounds, so proteinoids, which are abiotically produced polypeptides. And this was tested by Sidney Fox. He made these by dripping monomers onto hot sand, rock, or clay and allowing them to bond the way that they need to make polymers. The third requirement is concentration and enclosure of organic compounds. So the proteinoids would clump together to make protocells, which we just saw on the last slide. And these were able to maintain an internal environment different from its surroundings. And they did exhibit some life characteristics that we have today, which are metabolism, excitability, and self-replication. There are, let me see if I can say these right. Coacervates, which are drops of polypeptides, nucleic acids, and polysaccharides, which are sugars, able to self-assemble, and microspheres, which were protenoids in cool water. So these actually would have that selectively permeable membrane, so things could pass in and out of these protenoids. They resemble living cells that we see again today as membrane-like boundaries. They can take in or let out specific substances. They will cluster as different types of molecules, and reactions can take place more easily within these substances because they are bigger. And then the last requirement for the origin of life is evolution of growth, metabolism, and reproduction. Remember, metabolism, which we learned way back when, happens through glycolysis, and then reproduction is RNA, or reproduction goes to the first nucleic acid. So the first life on the earth were prokaryotic. So these were anaerobic, which means they did not need oxygen, which is good because our environment did not have oxygen in the atmosphere. And then heterotrophic, so they got energy obtained through glycolysis. These would multiply, and when that multiplication occurred, it increased the competition for nutrients because they all consumed the same thing. This led to the origin of autotrophs, and they made glucose by chemosynthesis in the beginning, and then eventually would translate that into photosynthesis. And, you know, we can look at and try to date things as best as science will let us with carbon dating, and we do that through fossils. So our fossil record is really important to try to, you know, see where things existed, because, you know, tectonic plates move over time, and when we think they occurred based on the amount of carbon left in the sample, those kind of things. So we first can see that our oldest evidence of the earth that, you know, we can try our best to date by are actually found in Australia by microfossils. They're about 3.5 billion year old prokaryotes, and the first life that we have, you know, proof of. And theories state that all organisms existing today descended from these. So the theory of evolution is that species change over time. Your autotrophs added O2 to the environment because they took in CO2 and, you know, released O2. And this made for an oxidizing atmosphere, which allowed for evolution of aerobic respiration to occur so that organisms could use oxygen for metabolism instead of just glycolysis. As this occurred, it would allow the ozone to develop, and that provides protection from the excess heat, which is what allowed our Earth to cool down. And then the terrestrial organisms evolved because temperatures decreased on the land so they could move, you know, from water to land. And eventually we know that, you know, we aren't prokaryotes, so eukaryotes had to form somehow. And they formed from the prokaryotes by endosymbiosis. So this is known as endosymbiont theory, which we, you know, don't have to discuss in too much detail. So your chloroplast and mitochondria were separate prokaryotic organisms with their own DNA, and somehow they became incorporated into other prokaryotic cells, which gave us that symbiotic relationship leading to those eukaryotes. So here is the endosymbiotic theory. You can pause this and read over that.